For any new viewers, my name is Rickard Anderkrans. I'm a healthcare analyst here at ABG Sundal Collier. Without further ado, I will leave the word over to Pekka Simula, the CEO of Herantis Pharma. So, Pekka, the floor is yours. Welcome.
Thank you, Rickard, and good morning, everyone. Great to be here. So, Herantis Pharma, and if we jump to slide 3 first for quick introductions on myself. So, I'm the CEO for Herantis Pharma, as Rickard said, since 2013, and you're also looking at a very happy CEO, even if also a very busy CEO. Just overnight, we completed a funding round, as announced this morning, so an accelerated book building process, and one is always very happy after a successful funding round, especially as we were able to bring in a new, very highly valued anchor investor in Sweden. But we move to the company itself. So next slide, please. Slide 4, Herantis Pharma. We are a publicly listed drug development company, listed both in Helsinki, Nasdaq First North Helsinki, since 2014, and then since last year, dual listed also in Nasdaq First North Sweden. And compared to many other drug development companies, we hold pride in 2 things in specific. One is our strong roots in science. So our work is truly based on leading scientific research in their fields in the world. And the second is that we aim at significant breakthroughs in our target indications. So we really want to change the way patients live with their diseases in our target indications. And the target indications are Parkinson's disease, where our drug candidate CDNF is in the clinic aiming to stop progression of Parkinson's disease, so aiming at true disease modification, which is the holy grail in neurodegenerative diseases. And then the other target is secondary lymphedema, where our gene therapy Lymphedema is aimed as the first curative treatment. And currently there are no approved drugs for the treatment of lymphedema. So again, this would be a real breakthrough. Both assets are now in clinical studies, in placebo-controlled clinical studies, approaching readouts well within the next year, so a very exciting time for the company. So next slide, please. Slide 5, just providing a summary. So neuroprotective factor CDNF targets stopping the progression of Parkinson's disease. So slide 6, please. Parkinson's disease is the second most common neurodegenerative disease, and it's currently treated with Parkinson's drugs, but unfortunately those drugs only treat the motor symptoms of the disease. They do not stop the disease from progressing. And Parkinson's disease is caused by degeneration of dopaminergic neurons in a specific area of the midbrain. And this degeneration continues despite the medication. So the currently available medication can only help the patient for a period of time, and then the disease progression will unfortunately make those drugs more or less useless. Parkinson's is also a significant societal cost. It was estimated already several years back that if there was a treatment that could stop the progression of the disease, such a treatment would save the societies almost $500,000 per patient in the United States. So when we're talking about disease-modifying treatments and our CDNF, we not only aim at alleviating the human suffering, but we also aim at helping societies manage the costs associated with the disease. Next slide, please. So slide 7, as mentioned, Parkinson's has drugs for its treatment. The current standard of care is still based largely on levodopa, which was discovered at the University of Lund by Professor Arvid Carlsson in the 1950s, or the preclinical work was done by him in the 1950s, which led to the clinical use of CDNF starting in the 1960s. So there hasn't been any essential breakthrough in Parkinson's for over half a century. And this is really where we hope to change the situation with our CDNF. So our CDNF is a natural human factor. It's a natural human protein that we all have in the brain. And it has an important role in neuronal survival. So its role is to help keep our neurons alive. And based on very compelling preclinical data, we have now proceeded to phase 1/2 clinical study with CDNF. The study is mainly funded by a European Union grant, a very highly competed Horizon 2020 grant. And the study is being executed at 3 excellent hospitals. So the University Hospital in Lund, of course, having great roots in Parkinson's therapies. Also Karolinska University Hospital in Stockholm, and then our local Helsinki University Hospital. And next slide, please. So slide 8 summarizes the mechanisms of action of CDNF. And very importantly, this is a multimodal approach. It's not just targeting one single phenomenon of the disease, like alpha-synuclein, which is currently very fashionable target in Parkinson's disease. Yes, CDNF also targets Parkinson's disease, targets alpha-synuclein, but in addition, it works through several mechanisms. And in the interest of time, maybe the most important one to emphasize is indicated by number 1 in this picture, endoplasmic reticulum stress, or ER stress. This is a cell-level phenomenon which is common to neurodegenerative diseases, not just Parkinson's, but also, for instance, Alzheimer's, ALS, and Huntington's disease. And it's a phenomenon in which the neurons start to lose their function. In the case of dopaminergic neurons, they stop producing dopamine, they withdraw their axons, and they start to proceed toward apoptosis, the programmed cell death. And CDNF has been shown to very potently protect cells from ER stress and help them recover from ER stress. Next slide, please. So this was signified first, of course, in several in vivo disease models, not just in Parkinson's disease, but also in other neurodegenerative diseases. And most importantly, as shown in this slide 9, in a primate study where Parkinsonism is due to a so-called MPTP lesion. So just like human Parkinson patients, these MPTP lesion rhesus monkeys have a severe Parkinsonism, which is caused by the loss of dopaminergic neurons. And if you look at the picture on the left, this indicates the severity of the lesion. So the dark staining in this picture shows dopaminergic neurons. And as you can see, almost all of the dark color is gone in this slide. So there's about an 80% loss of dopaminergic neurons. This corresponds to relatively advanced Parkinson's disease. But all of those neurons are not permanently dead. They have just lost their phenotype, their ability to produce dopamine. And when and we call these undead neurons, we call them Lazarus neurons. So if you look at the picture on the right, after 3 monthly doses of CDNF in these primates, we saw a tremendous restoration in the dopaminergic function. So as you can see, much more dark staining in the picture on the right. And this is the Lazarus neurons that we've been able to recover. We've been able to restore their phenotype. They are again connected to the surrounding neurons. They're again producing dopamine. And of course, if this was repeated in human Parkinson's patients, it would mean not only stopping the disease progression, but even reversing the disease progression. In these animals, this result very nicely correlated with improved gross motor function and fine motor function, and also for the first time in the world, as far as we know, in the non-motor functions. So the animals that were treated with CDNF also had a much less depressive behavior and much greater activity than the untreated animals or the animals treated with a positive control. So, and this is very important because Parkinson's patients say that for them, the non-motor symptoms are actually often even worse for their quality of life than the motor symptoms. And there are no treatments for the non-motor symptoms of Parkinson's. So, based on this, we believe that CDNF can address two of the main unmet needs in Parkinson's: disease modification and also non-motor symptoms in addition to the motor symptoms. Next slide, please. So, CDNF is currently in a randomized placebo-controlled clinical study, and this study has been fully recruited, and the study is broken in 3 parts. There's first, as indicated on this slide with the dark blue, there's a main study, which lasts for 6 months, 6 monthly doses of CDNF in Parkinson's patients. And in that part, the patients are randomized in 3 groups: a lower CDNF group, higher CDNF group, and a placebo group. This part of the study was unblinded in last February, and we already announced very exciting biological signals in the CDNF-treated group. But the study still continues, so it's currently in its extension study, another 6 months of CDNF treatment, and during this period also those who initially received placebo are now getting the active CDNF. And next slide, please. Current conclusions from the main study. So initial results after the first 6 months of treatment. Most importantly, this is a first-in-human study, so from a regulatory viewpoint, it's a safety study, and so it was great to see that there are no differences, essentially, between the side effect profiles in the placebo groups and the CDNF groups. So no safety concerns associated with CDNF at this point. But then, very excitingly, we are following these patients with a sophisticated Detta PET imaging. So it's a surrogate for synaptic density of the dopaminergic neurons. So it's basically a similar surrogate marker as the previous slides showed on the primate study. So a surrogate for seeing the viability of dopaminergic neurons. And in this endpoint, in one of the 2 CDNF dose groups, we saw 60% of patients responding to CDNF treatment. And among those were 2 patients, one had an over 50% increase in the Detta PET signal, and one had an almost 50% increase in the Detta PET signal. And naturally, or in the normal patient, average patient, it's known that this signal declines by about 6 to 10% every year. So in the first 6 months, we would have expected to see a 3 to 5% decline roughly in these patients. And instead, we are now seeing a 50% increase. That is very significant. Of course, we should keep in mind that this is a first-in-human study. These are just the initial readouts. So we look forward to the 3rd quarter of this year when we will have readouts from the full 12 months of treatment. But at the same time, we're also encouraged by the fact that these are advanced Parkinson's patients. These are patients with more than 10 years of disease history. We had to start with these, let's say, more challenging patients, definitely not optimal patients for a neurorestorative treatment, but the regulatory authorities always push for starting from this group. But if we jump to next slide, so slide 12, please. We believe that when we hopefully next can start treating earlier stage patients who have much more dopaminergic neurons left, as indicated in these pictures by the declining darker curve. So an untreated patient has a continuous decline in the dopaminergic neurons. In the top right corner, treatment starts at 10 years after symptom onset, so 15 years after the disease has actually begun and CDNF treatment started. We see that we believe we can to some extent increase the amount of dopaminergic neurons. But then if we could start the treatment 5 years after a symptom onset, the lower left corner picture, we believe that we could get retain the patient at a significantly higher amount of dopaminergic neurons and thereby a significantly lower level of symptoms of the disease. And this could really, we believe, change the rest of the life of the patient. Slide 13, please. So CDNF is a protein. It doesn't pass the blood-brain barrier, so it has to be administered directly in the brain, as we are doing with a sophisticated medical device, very similar to a deep brain stimulation device, a common treatment in Parkinson's disease. However, we'd of course like to administer CDNF easier. That would also could also enable its application in other neurodegenerative diseases where a medical device approach may not be as suitable. And here we are very excited about the early development of our X-CDNF, the next generation CDNF. These are fragments of CDNF that have the same activity, but that penetrate the blood-brain barrier. So we believe that the next generation could be dosed, for instance, as easily as insulin in type 1 diabetes patients. So a procedure that even a child can do. And this would, of course, be hugely exciting. And this X-CDNF program has already proceeded to lead optimization stage. So we are working our way toward clinical studies also with that. Next slide, please. Slide 14 just summarizes some of the essential comparisons or peers in Parkinson's drug development. There are not too many clinical stage assets with truly disease-modifying potential, and we believe that our CDNF compares very favorably among those, as it is a multimodal approach. So that summarizes CDNF, and in the interest of time, let's move forward with our other clinical stage asset, Lymphedema. Slide 15, as it says, gene therapy for curing secondary lymphedema. So if we move to slide 16, please. Secondary lymphedema is a disease that's caused by some compromise in the lymphatic system. So the system that carries lymph, the liquid, from the body, from tissue. So for instance, as in the picture, breast cancer treatments can damage the lymphatic system so badly that the lymph begins to accumulate in tissue. And in the case of a breast cancer-associated lymphedema, like in the picture here, the accumulation of lymph results in tremendous progressing and permanent swelling of the affected arm. If you imagine what life would be like living with an arm that's continuously swollen like this, you can guess that it's a very painful, disabling disease. It's also a very expensive disease for the societies. Again, it requires lifelong attention, as there are no efficacious treatments for the disease itself, only its symptoms. And next slide, please. Our approach is again very natural. So here we are harnessing another human factor, the human growth factor VEGF-C, which is highly specific for lymph angiogenesis. So it's the mechanism that naturally grows new lymphatic vessels in our body. So as shown in slide 17, if you look at the picture on the top, the green lines here indicate lymphatic vessels. The dotted line indicates a cut or damage across those lymphatic vessels. So when lymphedema is administered in that damaged area, then you reach the middle picture where new lymphatic capillaries start to sprout in the area, and then eventually the lower picture, those will mature into a functional lymphatic network. So we really aim at reconstituting the lymphatic system to help the body again circulate lymph normally. And next slide, please. So slide 18, where our development has reached a fully recruited phase 2 study where all treatments have already been completed. We're doing this in breast cancer-associated lymphedema, and the study is now in a 12-month blinded follow-up period, which will end at the end of this year. So first quarter next year, we will announce the top-line results. We're also already preparing a phase 3 study, so hoping that the readout from the phase 2 study will be strong. We look forward to launching a phase 3 pivotal study as soon as possible after the readout. And next slide, please. Lymphedema market is definitely a very significant market. It has long been a very underserved market, and still there are very few programs targeting lymphedema in the drug development world. But its awareness has increased very significantly thanks to Hollywood superstar Kathy Bates here in the picture, and thanks to the patient advocacy group Learn, very strong, especially in the US. Kathy Bates herself has breast cancer-associated lymphedema, and he's been a very important and vocal spokesperson for all patients in the world. In the interest of time, and hoping to complete in time, even though we were able to start a bit delayed, slide 20. Herantis' outlook remains unaffected by the pandemic. We were very fortunate in the timing of our clinical studies, so there were no essential impacts on the clinical studies of ours from the pandemic. And if we jump to the next slide, please. Expected news flow looks very exciting for the relatively near future. So in the third quarter of this year, we'll have the readout from the full 12-month treatment in our Parkinson's study. And in the first quarter next year, we'll have the readout from the phase 2 study with lymphedema. And last but not least, slide 22, please. So as we are moving forward toward later stage development, we also announced relatively recently that we have recruited a new experienced CEO, Dr. Greg Cook, who will join the company in July 2020. And I have promised that I will remain fully committed to the company. This is, of course, a very important development also for me. I've fully been endorsing and even to some extent pushing for this to ensure that the company is led by the best possible person going forward. So I really look forward to helping Greg get started in his new role. I think it's time to wrap up now. So we win back some of the time that's been lost earlier today. But if there's a question or two, I think there might just be time for that. Or Ricard, where are we?
Yes, we'll keep the Q&A session quite brief, but looking at, I took the freedom of opening up the final slide here, and just there might be some investors who are worried about the commercial potential for CDNF given its invasive administration. Could you add some flavor on why you believe there's still considerable commercial potential for this drug candidate? As you see, you have an estimated market potential of 1.6 billion euros in the slide. So could you please elaborate on that?
Sure. I mean, this was, of course, also a concern with deep brain stimulation when that started to emerge in the market. Funnily, no one still knows why deep brain stimulation actually works. So in advanced Parkinson patients, how it actually helps alleviate some of the motor symptoms in those patients when their disease has progressed quite far. And still, it's been a very successful market. So it's become a very significant market. And let's keep in mind that that's not even a disease-modifying treatment. It, again, just helps alleviate symptoms in late-stage Parkinson patients. That has shown that there is tremendous willingness in the Parkinson patients and clinicians to undergo an invasive procedure, even to just alleviate motor symptoms. So what we think that this is the strongest signal of willingness of the patients. But of course, we have also heard it from a lot of the patients that, who say that once they've lived with Parkinson's for a couple of years and they understand how it's going to impact the rest of their lives, many of them say that, you can drill any number of holes in my head if you could only stop the disease from progressing. And there's a significant difference between, of course, CDNF and DBS. CDNF could truly stop disease progression if it works as well as we believe.
Excellent. And just as a very brief final question for any investors or potential investors who might not be aware, will you be trying to market these drugs alone, or what's the commercial strategy, just very briefly?
Well, in fact, we believe that we could bring this to the market on our own. It's, Wilson Therapeutics showed a great example there, that if you have kind of a niche therapeutic targeting a specific hospitals, for instance, then that's something that you could take into the market alone, at least in some markets, such as the US. CDNF and Parkinson's disease certainly is a huge undertaking, even for many mid-sized pharma companies. So I think that our smartest option would be to find the right partner at the right time.
Excellent. Thank you very much for your time, Pekka, and we'll move on with our busy schedule. Best of luck going forward.
Thank you, Rickard.
Thank you. Take care. Bye.